Okay. Very good evening. Hello.
Good evening, sir.
Good evening. Good evening. So today is the last class for our syllabus. Okay.
Yes,
So that last class stellar evolution is over. Right?
yes,
Or
sir.
is explained stellar evolution? Types of the different types of the steels in Pteridophyta. Haplosteel and diplosteel, actin steel. I think this one is completed.
Yes, sir.
Is right? Okay. So now today we discuss the remaining two units are there, heterospory and seed habit. So another one is some fossils are there, fossil type studies. We can discuss that one today. These two units are Xylotum and Rhynia. Another third one is the Calamatis. These three are there. Is okay clear? So okay, is audible?
Yes, sir.
Okay, thank you. So what is heterospory? So heterospory is a condition that interprets the production of spores of the different sizes and different developmental pattern also. Two types of the spores are produced in single species. In a single Pteridophyta, that one we call as a heterospory. So two types of the spores are produced. So in the previous class we discussed that one, heterospory. Selaginella is produced with the heterospory, produced the different types of the spores. So here the two different sizes, which are the small and another one is the large spore. So these small spores we call as a microspores, and larger one termed as a megaspore. They are the developmental pattern is also different. The microspore germinate to produce the male gamete, which bear the male reproductive organs, that is called as antheridia. And megaspores, they are produced the female gametophyte, which bear the archegonia. That one is nothing but female reproductive organ. So here, two types, the micro and mega spores are produced in the single plant. These two kinds of the spores are produced in two different sporangias are there. Single plant only, but different sporangias are there. Microsporangia and megasporangia. Microsporangia is produce microspores, megasporangia to produce the megaspore. Okay. So that is the microspores are produced in large number or small in size, but the megaspores are is very less in number and large in size. So here the large size of the megaspores is attributed to availability of more nutrient. They have few in number for the megasporangium. So this reduction of the number of megaspores has been achieved either through the reduction in number of spores by the disintegration of the development of spores. So due to the some nutrition or nourishment of the remaining megaspores, some are the degenerated and remaining are developed as a megaspore. Okay. So what is the, for example, Selaginella, Isaitis, Marsilia, these Azolla, these all are the heterosporous plants are there. Okay. So what is the importance of heterospore? So this microspore gives to the microgametophyte bear the male reproductive organ. Megaspore develop into megagametophyte. That one to give the female reproductive organ. So there are the the difference in size of the spores is directly released to the reproductive gametophyte. So the heterospore is and indicate the sex determination. Is clear? So heterospore to produce the two types of the spores are there. Microspores, another one is megaspores. So microspores to give the male gametophyte, megaspores to give the female gametophyte. So that's why here sex determination is there. Sex recognition and sex determination is present in the heterospore. In the homosporogen is clear? Okay, that is it. Understood? Okay. So in the homosporous genera, the sex determination is observed in the gametophytic stage, but the heterosporous genera is observed in the sporophytic stage. By observing the detail of the development of Selaginella, it becomes clear the sex determination starts during the sporogenesis. So, because of the heterosporous plants, they became free to grow in the varied environment condition, which may be happen during the course of the development. So, the heterospory is important because it is ultimately leads to seed the development. Because the heterospore is gametophyte tissue to reduce their in reduction in its endosporic nature partial or sometimes it is complete retention of the gameto or megasporangium and female gametophyte on the sporophyte. The heterosporous plants show the reduction in the number of spores. All these characters show tendency toward the seed habit. So in the heterosporous plants, Selaginella is the megaspore having female gametophyte derive its nourishment from food made by sporophyte. So it is remained independent from all the external factors which may be normally provide headness in the growth of the free living gametophyte. So what is the origin of heterospory? It is clear that heterospore is originated due to the reduction of the spore number in some sporangia due to the availability of more nutrient to the remaining spores. They increased in the size. So Rashid, he studies the eczytem in the Ceratopteris and Platysoma is mentioned in that. Is the heterothallism may have produced the heterospory in evolution. Heterothallism also one cause to explain the heterospory. It is explained in the eczytem and Ceratopteris homosporous but produce two types of gametophyte, which are the smaller spores, one to produce the male gametophyte, larger one the female gametophyte. There is the plants in eczytem and Ceratopteris and Platysoma. There is different incipient heterosporous forms are there. So Duckett and Pang is studied in the sexual behavior of the Platysoma microphyllum and Eczytem giganteum. So according to them, the sticking similar in the sexual behavior of Eczytem and Platysoma suggested that the crucial step in the development of heterospory is an intimate association between the gametophyte morphogenesis and sex organ formation. The gametophyte, the datian and dimorphism resulting in the form this association may have preceded the heterospory in evolution. The origin of heterospory can further be decided through the evidence from paleobotany. So some evidence also there to study about this heterospory. So through the fossil studies, it is clear that widespread heterospory was existing in many genera of like Lycopodium, Synapsida, and Pteridophyta members. So they have common in late Devonian and early Carboniferous period. The importance of heterosporous genera during this period were Lepidocarpon, Mesocarpon, and Sigillaria strobus, and Calamocarpon, Calamostachys, Peleostachya. These evidence also indicate that some of genera have reached up to the seed stage. So this is advanced to the heterosporic condition. So according to William and Scott, Calamostachy and Calamocashina from initial stage of heterospory, and Calamobeniana is a homosporous condition, but some sporangia had unequal size spores are produced. In Calamocashina, the micro and megaspores were present separately, and some abundant spores were also present among the megaspores. So Kaliner is represent the aborted spores among the megaspores of Staropteris and Leptocarpon, Thalamocarpon. So another scientist is Arnold. Also reported presence of megaspores in Leprostrobus. All these evidences from fossil records indicate that the heterospory occurred quite early in the plants due to the abortion of spores in the sporangia. So these environment conditions also play an important role in the formation of the heterospory. So another evidences to developmental stage during the heterosporous, so means the developmental stage of the heterosporous pteridophytes may also some important in the origin of the means heterospory condition. So here, what are the demarcation here? At the time of the sex determination, nature of the archegonia means archisporial cells in the archisporium in sporangium. So number of such sporocysts which is initiated and complete the process of meiosis. Number of spores which are ultimately occur at maturity, and the origin of tapetal layer and time of its disaggregation. So considering the sporangial developmental stage in Selaginella, it became clear that the development up to porosis stage is similar in both microsporangia and megasporangia. In microsporangium, all the sporocysts divide meiotically and form a large number of microspores. But in the case of the megasporangium, all the sporocysts is degenerated except one, which is undergoes to the meiotic division and form four functional megaspores are there. In some species, these four megaspores are in different dimension, which are either in the that is the Selaginella erythrosporus. Only one functional megaspore is present in megasporangium. So Rashid is observed some large size of the spores in microsporangia of some species of Selaginella, showing an evidence of the origin of the heterosporus. In Marsilia, there is no difference between the microsporangial or megasporangial development before meiosis. In the microsporangium, all the 64 spores survive, but megasporangium only one out of the 64. In the case Salvinia also, this one we can see. The developmental stage in the genera of Selaginella is Itis, Marsilia, and Salvinia, which is indicated there is no generalization of the origin of heterospory. In some cases, it starts at the pre-meiotic stage, while all others, the heterospory originated only at the post of the meiotic stage. So, experimental stage, here the Selaginella and Marsilia, they are the nutritional factors play an important role in the origin of heterospore. Okay. So heterospory array. So here some stages are there to formation of the heterospory. So formation of two types of the spores are there, that is called as a heterospory. A reduction in number of functional megaspores and the retention of megaspores in megasporangium for the reception of microspores or pollen grains elaborated in the megasporangium. Fertilization and formation of embryo takes place, which is the in situ condition, and the formation of integument due to the histological union of megaspores and megasporangium. Seed habit in Selaginella. The Selaginella shows the seed habit because it shows two types of the spores, that is heterospory. It shows two types of the spores are there in Selaginella monospora and Selaginella in the repertories. So number of megaspores is reduced, one which is conformed to seed habit, and the germination of the megaspore starts with the megasporangium, but is the germination and time of retention with the megasporangium different in different species are there. So in Selaginella repertories, the Archegonia developed in situ conservation. So in Selaginella repertories, is fertilization and embryo it developed take place within the megasporangium. So according to Parihar in 1977, this is the certain species of Selaginella suggested that clearly the evolution of seed habit. These Selaginella shows do not to considered as the ancestor to the seed plants. So reasons for the absence of well-developed seeds, these Pteridophyta members. The reasons for the absence of complete and well-developed seeds in the Selaginella and other species as follows. The absence of integments around the megasporangium. The except in few species there is absence of permanent retention of megaspores within the megasporangium. The complete histological fusion between the megasporangium, well, megaspore is absent. Okay. So in this heterospory and seed habit, heterospory is sometimes is asking essay. Okay. Okay, clear?
Sir.
Mm. They are asking sometimes as a essay question. Okay. So the next one, some pteridophytes. Okay, clear? So the fossil, some pteridophytes we are to discuss here. That is the remain plants of animals which are lived in the remote past and preserved in the rock in the form of the fossil. This is called as a fossil. So a little knowledge on the fossil record of any group of organism is essential to know the types of the lived and idea of evolutionary process. The discovery of new types of the fossils, which is useful in prepare the continuation story, the development of plant kingdom from more than 1 billion years to the modern time. The diverse types of the plant group have been that is the recorded. Modern botanists can know never about such a plant group that is dominated and the earth crust in the past. So the fossils can throw light on the origin of the most present day group of aquatic plants to the terrestrial situations. So the fossils record also help to know about the change in the earth climate and their effect on the flora fauna. So there are the variety of the forms of the fossils is depending upon the condition under the fossilization process. So in this one, so Rhinia. So these Rhinia, the division is Xylophyta, Xylophytopsida, and order is the Xylophytales. Family is the Rhiniaceae, and genus is Rhinia. So here Kidston and Long discover these Rhinia. So this Rhinia sometimes when asking the short question, Rhinia or Xylophyton and Calamity. This Rhinia is majorly asked as a short question in your exam point of view. Okay, clear?
Yes, sir.
Okay. So the genus is named after locating Rhinia in the Scotland area. So the Rhinia is represented by two fossils are there, Rhinia gigwani and Rhinia major. These two species are there. Okay. So what is the morphology of these Rhinia? So Rhinia gigwani was the small herbaceous plants about 20 centimeters height. It is consist of slender cylindrical aerial stem of the about 2 to 3 millimeter diameter and branches rising from rhizome. So rhizome is consist of the turf of rhizoids on the underside. The rhizoids perform the dual function, which is the anchor and absorb, means which is to fix these stem into the fixed soil and to absorb the mineral and nutrition. Okay. The aerial stem was dichotomously branched and taper gradually toward the apex. The stem bear the number of hemispherical outgrowths which are considered as rudimentary leaves. The aerial stems may be end in the pointed tip. They bear the some oval-shaped sporangias are there. Stomatas are present in all over the surface of the aerial stem. In the advantageous to shoot branches also arise from the aerial stems. Okay, so in this Rhinia, they are asking short question morphology is compulsory. Morphology and sporangia. You should write the sporangia structure in the short question. So in the marker in 1959 is considered as the present in the gametophyte and the upper aerial part as a sporophyte based on the following evidences. The rhizome possesses some flask-shaped cavities which were considered as a disintegrated reproductive organ. So some specimens from the Munich consist of some groups of 4 cells with the opening in the center body and resemble the surface view is embedded in the archegonia. So these two evidences So there are the two evidences support the concept of the rhizome and subterranean creeping axis as the gametophyte. So here the presence of hemispherical outgrowth is the erect stem may be considered as a set of the formation of additional branches. The additional branches were actually the divide of the vascular connection within the main stem. They often became detached and afford a ready means of the vegetative propagation. Rhynia major is another species which is larger than the Rhynia guinea vagini. This is an distinct guinea vagini is a large size in the hemispherical outgrowth and adventitious branching, large size of the sporangia and spores. Another one is the internal structure of the rhinia. When we take the transverse section of the aerial stem, revealed as a distinct epidermal layer is interpreted at the places by the presence of stomata. So this epidermal layer was covered by a thick cuticle. The cortical tissues was well differentiated into outer and inner cortex. The outer cells is the few intercellular space, but the inner cells are smaller one and there is more intercellular space. The inner cortex is probably possesses the photosynthetic pigment and intercellular space are connected with the stomata through this some space between the outer cortical cells. So in the center, center stem was occupied by the vascular stand of a simple organization. The brackets are present at the center, which is exhibit the annular or sometimes spiral thickness. The inner brackets are smaller than peripheral brackets. A, E, which is exhibits the annular or sometimes spiral thickness. In inner brackets are smaller than the peripheral bracket, which is indicate the endark type of the xylem tissue. The cells is surrounding the xylem and smaller without interstellar space, and it is considered as a phloem. There are no traces of sieve plates and four in the phloem. The phloem of Rhynia is indifferentiated, that is the living Pteridophyta. Sieve tubes and phloem parenchyma, which both are present in Rhynia. The pericycle and epidermis is absent. So in the rhizome, the cortex was differentiated into outer and inner cortex. The xylem consists of very brackets are there. Okay. So what is the your life history? The rootless and leaflets plants was the dichotomously branched aerial shoot. That is the aerial shoots possess the terminal sac-like structures are there. That one we call as a sporangia. This one is known as the reproductive uh, type, organ in the fossil taxa. The sporangia are the club-shaped or cylindrical structure about the 4 millimeter long and 1.5 millimeter broad is there. This was here the outer epidermis and middle thick layers are there. The inner wall is called as the tapetum cell. So these tapetum cells to act as a nutritive or nourishment layer we call as a tapetum layer. So on the outer level is which was cutinized, the outer one is heavy cutinized layer is present, and the inner sac or cavities consists of several spores arranged in the tetrahedral manner. So there are the homospores condition is there. So these spores are roughly around 40 microns in diameter. The development of the megasporangium was eusporangia type. It is developed from the group or row of the superficial cell, which is divided the pericanal developer to form the outer primary wall layer and inner primary sporogenous layer. So, as a here paleobotanist interpreted that the flask-shaped cavities present on the subterranean creeping axis as disintegrated as a reproductive organ, that is the antheridia in some fossil groups. Okay. So here in some fossil groups there are four found in the groups of the four cells with the openings in the center and considered them as rudimentary archegonia. So they assume that the Rhinia species are primitive plants, hence they possess such mass where embedded sex organs are there. So based on the observation of Rhiniaceae, can be treated as a synthetic group of the plants is containing the characters of vascular cryptograms and bryophytes. So on the other hand, the retaining some characters of the thallophytes and other side. The scientist is believed that there is an environment were quite unfavorable and their plant body was reduced due to the environmental pressure. So next one is the Xylophyton. So the division is Xylophyta, class is the Xylophytopsida, order is the Xylophytes, family is the Xylophytaceae, genus is the Xylophyta. So is very important member of the Xylophytes. The plant was reconstructed from the fossil bits was observed in the one from the lower Devonian rocks of the Gaspé, Canada. The name Xylophoton is a princeps was given this reconstruction. So now it is discovered in certain other parts. The Dobson is pictured in the Xylophoton princeps was having upright fork stem of the somewhat is slender proportion is springing from creeping rhizome. The aerial structure stems were ornamented with the spines and upper fertile branches was divided them. The tips of the younger branches were curved in the succinate manner, similar to the ferns. The stem was less than 3 feet in height and varied in the diameter from 1 to millimeter. So these spines varied from 0.5 to 2.5 millimeter in length. These spines have been regarded as a foliar in the nature by several authors. The arbor compared them the hemispherical outgrowths of the stem of the Rhenia Guinea vagini. In the surface of the spines seems to cover the cuticle and epidermis. So aerial branches was dichotomously forked. Branching was profuse as compared with the Rhenia. The sporangia were borne terminally in the pair on the fertile aerial shoots. The sporangia were long, oval, sac-like structure, much similar to those Rhenia. So the length of the sporangium varies from 4 to 5 millimeters. In breadth, which is 1 to 1.5 millimeter. The exact structure of the sporangial wall is not visible, but it seems to be compared to two layers. The spores are arranged in the tetrads and have more or less in smooth wall. Is around the 60 to 100 millimeters. They are all the same in size. Some evidences as the anatomy of rhizome was available. So Dawson is take this section to similar to the idea. The Edwards in his unpublished work was described the epidermis was stomata were more numerous in Rhenia. There are also evidences that called so two groups of cells are elongated stomata. This shows that remain set of the photosynthetic in the outer cortex as in Rhenia. There are other species of Xylophoton was being discovered from other part of the world. One other the Xylophoton Veminensis was found to be at the tooth bottle. So Wyoming and USA in the lower Devonian rocks, but out of the other species that is Xylophyton princeps has been only to study the details and the such lack of the information in their structure. So another fossil types from the Pteridophyta, that is the Calamatis. And the division is the Spinophyta and the class is Calamopsida. Order is the Calamates. Family is the Calamates family. The genus is the Calamates. Good. So Calamatis and its relatives existed at the peak of the development of upper Carboniferous along with the Lepidodendrons. Found on the coal mines and swamp forests, and they became extinct in the end of the Permian period. So these calamities was one of the prominent representative of the calamities and was reported in the lower carboniferous rocks of the Pitcairn and Scotland. So stems was rigid and the ridges alternating successive internodes. Xylem is similar to the equisetum. So as per the protoxylem tends to break down to the form central axis. So the secondary wood was laid down to the outside of the metaxylem, but the primary wood is raised in the wide with the appearance of having separate stands. But the primary wood race was formed from the constitution continuation of vascular cambium. The height of the calamities is not clearly, we know that because of the fragmentary nature of the remaining. However, it is estimated around so the diameter of trunk was around 30 centimeters, and stem is exhibits rides and the growths are there. So the genus Calamities is divided into three subgenera are there, which is based on the branching pattern and nature of the ridges and growth. That is Eucalamities and Mesocalamities, Stylocalamities. These three groups are there. So Eucalamities here two branches at every nodes. And another one is Mesocalamate. Nodes are different, ridges are alternating, and sometimes is continuing state across the node. Stylocalamities will branch only near the top of the erect origin and pipe-like trunks are there. So based on these arrangements of the branching here, there are the three groups are there. The leaves were unbranched with the single midvein and occurred in the whole of the four to six. They were free on the base of the thumb. This shows some fusion into sheath. So, these cones in this calamities was born on the different ways. Some bear the cones at the node and other may be terminal part of the branching. They are consist of there are oral sporangias bear the four respectable sporangias are there. This number of bracts and sporangias spores were varied in different species. Calamities as homosporous condition, and some species also recognized as a heterosporous condition. The number of calamities bring out some interesting evolutionary trends which are parallel to the number of lepidodendrons. The production of increasing amount of secondary xylem and reduction of the primary wood in to these two groups. The sporangia was protected by the overlapping sporophylls in lepidodendrons, while the bracts are calamities also another interesting character in the lepidodendrons. So these both groups are the extinct nowadays. Okay. from this class, it was completed in our portion. Okay, clear? So,
Sir,
Raini,
selective.
sorry,
Sir, can you provide a selective question, sir?
important questions.
Sir, sir.
So already when I am taking the class, already told no, that important topics. From that
Yes, sir.
here, so from that bryophytes, general characters of bryophyte, they are asking sometimes. Another one is the Markantia sporophyte, Anthocera sporophyte, they are asking. Markantia, Anthocera sporophyte. And evolution of sporophyte in bryophyte, compulsory asking question. Evolution of sporophyte in bryophyte. From the Pteridophyta classification, Pteridophytes classification, and Lycopodium structure, Salaginella equisita. These type studies means life cycle and type studies they are asking.
Sir, please, every topic important questions. Tell me.
No, already from that bryophytes onwards, I telling no. Lycopodium Salaginella.
Bryophytes and sporophytes, sir.
Evolution of sporophyte in bryophyte. Compulsory asking that question.
Next, sir.
So from that bryophytes, so general characters of Bryophyta.
General characters of Bryophyta.
Bryophytes. Then another one here, 12th unit, Markantia type study.
Markantia.
Type study, life cycle, total life cycle. They are asking
Total
sometimes.
life cycle types. Next, sir.
Then Anthoceras also. Sporophyte.
Anthoceras.
Sporophyte. They are asking.
Sporophytes.
Okay. Evolution of sporophyte. Evolution of sporophyte in bryophyte.
Evolution of sporophytes in bryophytes. General characters of Bryophyta, Markantia, total life cycle and general characters.
Life cycle also means sporophyte development.
Anthoceras and sporophytes.
Yes. Anthoceras sporophyte.
Next.
Pteridophyta classification.
Pteridophyta classification.
Pteridophyta classification. And another life histories of Lycopodium, Salaginella equisita. They are asking individual question, but these three are the important.
Salaginella.
Lycopodium equisita.
Lycopodium.
So major times they are asking the Lycopodium, but sometimes only equisitum also they are asking.
Okay, sir.
Then stellar evolution.
Stellar evolution.
Stellar evolution in Pteridophyta. In short question, stellar evolution. Sometimes asking short question, telome theory also.
Telome theory, okay, sir.
Telome theory. And another heterospory and seed habit.
Heterospore and seed habit, okay.
Heterospory and seed habit is also asking. And these fossil pteridophytes also asking short questions, Rhinia and Calamatis.
Okay, sir. Okay.
Rhinia and Calamatis major times are asking. Okay. From that algae, first unit and black one. Black, from that algae, black one and black two.
Sir. Algae.
Black one and black two.
From that black one and black two, here thallus organization. Thallus organization of algae.
Organization of algae. Okay.
Thallus organization, algae. Algae. Thallus organization. Major times they are asking.
Okay.
Okay. Some life studies, volvox. You can see the volvox and Polycyphonia ectocarpus. Also, sometimes they are asking.
Okay, sir.
From algae, you read the life cycle.
Volvox ectocarpus. Other ectocarpus.
Ectocarpus also you can see Polysyphonia. You see that
Oh.
one. Okay. Another one is the Synophyta character. Synophyta characters. You see that. And another one, economic importance of algae. Compulsory asking basic question.
Okay, sir. Okay.
Economic importance of algae.
Okay.
These some points, some questions. So you focus that one for the preparation.
Okay, sir. Thank you, sir. Thank you so much.
Exam point of view. These questions compulsory coming in your
Okay,
exam.
sir.
Okay.
Okay.
So you stay sometimes. After that, we leave the class. Okay.
Okay, sir. Okay. Thank you, sir.
You stay sometime.
Thank you, sir.